Have you ever wanted to learn about CHARD syndrome and how it develops? Then you are in the right place. CHARD syndrome is a rare genetic syndrome that is recognized by its key physical features. The name CHARD syndrome was created collaboratively by a group of geneticists in the 1980s to create the mnemonic that describes the physical characteristics that defined CHARD syndrome at its time. The mnemonic is as follows: colomboma, heart disease, atresia coloni, restriction growth and development, genital hypoplasia, and ear abnormalities. Although these features are no longer used in making a diagnosis of CHARD syndrome, the name remains unchanged. Throughout the 1990s, scientists began to study the physical characteristics of CHARD syndrome. As technology progressed, scientists were able to better understand CHARD syndrome at a cellular and molecular level. Scientists asked the question, what causes CHARD syndrome, and tried to understand the mechanisms behind it. In 2004, a paper was released entitled Mutations in a New Member of Chromodomain Gene Family Causes CHARD Syndrome. This paper showed that there were changes in the DNA code for the protein CHD7 in people with CHARD syndrome. This change in CHD7 gene altered CHD7's function within the cell. When CHD7 can't do its job properly, CHARD syndrome is the result. To understand how CHARD syndrome affects fetal development, it is important to first understand the basics. For a cell to stay alive, it must use the genes in the DNA to make RNA. RNA then must be made into protein, which will do the work to keep the cell alive. DNA is over 6.5 feet long, but it needs to be stored inside the cell, which is smaller than a grain of sand. But how do you put an over 6.5 foot piece of thread into a grain of sand? Just like a thread wrapped around a spool, DNA wraps itself around individual structures of histones. Like chapters in a book, 8 of these histones come together to make a book of genes called a nucleosome. The nucleosomes continue to compact together to create a library of genes called a chromosome. Humans have 23 different pairs of chromosomes. This lets the cell have 2 copies of every gene. 1 copy is passed down from one parent, and 1 from the other parent. Our cells use both copies of each gene to make sure that we have enough proteins in our cells to be healthy. Sometimes there's an alteration to the gene, which makes it not work properly. This can cause proteins that are made by this gene to be misfolded. Misfolded proteins are not healthy for the cell and can cause damage to the cell. In CHARD syndrome, CHD7 proteins are misfolded, and then CHD7 cannot do its job properly. CHARD syndrome can be inherited from one parent passing down the non-functioning CHD7 gene to their child. Many individuals with CHARD syndrome have no previous history of an abnormally functioning CHD7 gene. This means that both parents typically have 2 functioning copies of the CHD7 gene, but the non-functioning CHD7 gene is created during pregnancy. The non-functioning CHD7 gene is an identifier for CHARD syndrome. How does this non-functional CHD7 gene impact the growth of a child during pregnancy? Every human starts out as an egg with 1 cell. Through the division of the original cell, the egg is able to create limbs and organs to become human. To do this, proteins need to be made at a specific time with a specific amounts of proteins. if there are changes to the time or the amount of proteins being made, then there are developmental differences. CHD7 is a very important protein in development. The CHD7 protein is able to control when proteins are made by telling them when to turn on and off the production of other genes. You can think of CHD7 as being the project manager of a crew building a home. Let's see CHD7 in action. Like mentioned before, our chromosomes are like a library of genes. Our nucleosomes are like the books within our library, and our histones are the individual chapters with the blueprints to make proteins, also known as our DNA. CHD7 must first find the genes it needs to make by identifying which histone it needs to open up to allow access for DNA. Once CHD7 identifies the histone it has to open, it attaches to the histone and temporarily removes it, just like taking a book off the shelf in the library. This causes a small section of DNA to unwrap so that the blueprint for proteins are accessible. Once the DNA is unwrapped, special proteins come in to translate copies of DNA. These copies are called messenger RNA. Messenger RNA is a copy of instructions to make each unique and specialized proteins. These copies of instructions are able to move around the cell and allows the original copy of the instructions to be safe for continuous use. CHD7 is not believed to be involved in the process of making messenger RNA. When messenger RNA is finished being produced, CHD7 must come back to rewind the DNA into the nucleosome. Essentially, CHD7 has to make sure that the book is placed back into the proper space to be used again. To do this, CHD7 will place the histone back into the nucleosome by rewinding the DNA around it. This causes the creation of proteins to be stopped. CHD7 can always reopen the histone when more protein is needed for the cell. CHD7 is needed to make different kinds of proteins. Without properly functioning CHD7 proteins, the cell does not have access to other proteins it needs to make limbs and organs, ultimately causing CHARD syndrome. It is important to realize that scientists are only at the beginning of the journey of understanding how CHD7 impacts fetal development and how it explicitly causes CHARD syndrome. In only 20 years, there have been many great discoveries, and we have many more discoveries to look forward to in the future. In the meantime, realize that the genetic alterations behind CHARD syndrome are not something in our control, but there is still plenty we can do to help support individuals with CHARD syndrome. While scientists work hard to help us better understand the genetics behind CHARD syndrome to bring us closer to future treatment, work from others has helped us realize that with proper supports, individuals with CHARD syndrome can lead fulfilling lives.